Merci pour cette belle invitation, pour cette sélection. C'est un grand honneur d'être ici avec vous aujourd'hui, de vous présenter le lauréat BONEV. Juste quelques mots avant de lui donner la parole. Notre équipe travaille à la régulation du développement et notamment l'épigénétique, le rôle de l'épigénétique et de la mémoire des états cellulaires dans la régulation du développement, ainsi qu'on a découvert récemment que certains phénomènes épigénétiques peuvent être transmis aux générations successives. Mais ici, dans ce contexte, en fait, cette étude vient du fait que depuis quelques années, on avait identifié le fait que l'organisation tridimensionnelle des chromosomes dans le noyau peut jouer un rôle important dans la régulation des gènes et dans la régulation du développement. Actually, I will switch also to English because also Boyan is going to speak in English anyways. I heard that we are on YouTube. Is that true? I don't know, but maybe the YouTubers will like it in English then. So just to say that this is a collaboration with the Tanai Lab in the Weizmann Institute in Israel, and they really did an important work here. And it was a great pleasure to work with them. So just to, no, that's not working. Just to tell you very briefly, we have a lot of DNA in our cells. We have, 2 meters of DNA that have to be compacted in 10 microns in a nucleus. So it's a tremendous amount of compaction that you need from the linear size of DNA to fit into the nucleus. And not only DNA fits in the nucleus, it has to work there, it has to be transcribed, it has to be replicated, repaired, to recombine when you have sexual reproduction. So you have to have a fine regulation of all these processes simultaneous with a fitting of the material, genetic material in the nucleus. So we know that this is done by a hierarchy of structures. So the first thing that happens is that DNA is compacted in nucleosomes, and these nucleosomes are basically proteins around which about 150 base pairs of DNA are wrapped around. And then we have a series of structures that end up forming chromosomes. And actually, this is what an interphase nucleus would look like. Chromosomes are more famous as sort of rod-like structures, but in a nucleus, as it's functioning, transcribing genes, each chromosome occupies a discrete territory. But what are the structures in between these two? Because actually forming nucleosomes is by far not enough to fit the DNA. You have to fold it another 1,000 times if you want to have DNA in the nucleus. And we know also that structure has not just a role of making the DNA fit, but also a regulatory role. So you have different levels of implications between the fact that you have spatial proximity of regulatory elements and you're going to regulate gene expression. But also you have to regulate, as I told you, replication and repair. And so we know that specific elements are close, but it might also be important where a gene is located geographically in the nucleus, what is its radial position, and also is there a topological organization? Are there domains which exclude other domains? And are there anchors, specific anchors that define these domains? There's a technique that was actually developed about 10 years ago that allows to study these events in high resolution, which is called Hi-C. And this is basically a technique based on cross-linking. So what you do is, if you have contacts in the nucleus somewhere, so this would be a nucleus with chromosomes, if you have a specific contact and you put formaldehyde, you can actually freeze this kind of contact. And then you can solubilize chromatin by a resection enzyme. And then you use tricks like putting biotin, that I maybe not go into details, use a ligase to trap these contacts, and then you can sequence them and produce maps. And actually, 5 years ago, I came to this very same place to show a work where we had done this in Drosophila, and that revealed the organization of Drosophila chromosomes. But of course, it's very important to do that in mammalian cells. And when you do that, with this technique, you can see that chromosomes form territories. I mean, that I skip over this because also microscopy can do it, and for reasons of time. But you also see these domains, okay? These domains here that you see. So this is a map where you have each place is a position along the chromosome, and the color is telling you how much contact you have between different points. And so these regions here are sort of domains that you see in different colors, and some of them might be active and others inactive. And if you zoom again into one of these domains, you might even see finer structures like this dot here, which could be the base of a loop, okay, one of these interactions. And the question is how many of these do we see, how dynamic they are. And to do this, we have to have very high resolution, and that's what the role of this project was, to provide the community with extremely high resolution in a well-defined differentiation system. And with this, I will close and give the word to Bojan.
All right. Well, first, I'd like to thank everybody here for coming, and also the Academy for this great honor, and Giacomo for the nice introduction. So the central question that we actually want to address is how is genome organization changing during development, and whether these changes actually have any meaning for gene expression and for cell fate. And the way that we decided to address this is by using neuronal developmental system. And this is not only because the cells undergo a profound change in what genes they actually express and in their morphology, but also because how the brain is organized is one of the central questions in human biology. And in order to study this from developmental perspective, we utilize two different systems, so an in vitro system and an in vivo model system based on mice. So in the in vitro system, what we are studying is we start with embryonic stem cells and we differentiate them sequentially into two stages, so neuroprogenitors and cortical neurons that you can see here. And importantly, we used FACS purification in order to make sure that we purify specific populations of the cells, so the cells that express the right marker for the correct cell type, but also so that they're uniform in their cell cycle, because the cell cycle is actually known to have a profound effects in chromosome organization. And in the other complementary approach, we actually use transgenic mice, where we cross two transgenic lines, so one which expresses a green fluorescent protein in the neuroprogenitor cells of the brain, and another one which expresses a red protein in the neurons of the brain. And by combining these, by crossing these mice, we can isolate the cortex of the embryos, and we can purify green cells that are not red. This will represent essentially the progenitor cells in the brain, and red cells that are not green, this will be actually the neurons in the developing brain. So as Giacomo mentioned, in order to understand how genome organization contributes to cell fate, we need to study this in the context of the other processes in the nucleus. So we need to understand how it connects with linear transcription marks, like for example by using RNA-seq, but also with the linear chromatin marks that are well known to control cell fate in the epigenetics processes. And we also developed a novel method because doing the Hi-C approach in vivo with very limited number of cells was challenging. So we have further optimized the technology, which we can now use to look at very rare cell populations using in vivo. And as Giacomo mentioned, we achieved the highest resolution up to date, which really allows us essentially to study how different regulatory elements connect with each other and whether they can contribute to cell fate specification. So the first thing that we examined is actually how are the different domains in the genome changing during neurodevelopment. So what you're seeing here are the domain boundaries that Giacomo mentioned about, and we asked the simple question of whether those boundaries between the individual domains are changing during development or whether they remain static, as has been suggested before. And what we found is that actually a very small minority of them, so only about 10%, undergo changes, but the ones that change, we can quantify the insulation of those regions, and we can see that there are two particular cell types. So those are the first type is boundaries that have high insulation and are then become reduced in insulation and essentially lose their insulation potential. And on the other side, we see boundaries that have no insulation potential in the first cell type, so in the embryonic stem cells, but then later become highly insulated. So then we ask the question, Does transcription play a role in this formation of the boundary? And what we found is that if you look at the genes that are in the neighborhood, so in the immediate vicinity of these boundaries, what we find is that when you have an increase of insulation, so when you form a new boundary between two different domains, what you see is an increase in gene expression and active chromatin marks at those regions. Conversely, on the opposite side, when you lose insulation here, what you see is a decrease in gene expression and again a loss of active marks, pointing to a correlation between transcription and insulation potential. So here I'm showing you one example at the developmentally regulated region. So what you see here, this is the linear genome, and this is essentially all the contacts that are mediated by the different points along the genome. So strong contacts are denoted by red, and insulation is denoted by blue. So what we see is that in an embryonic stem cell where this gene is not expressed, the contacts around its promoter are fairly uniform. On the contrast, and to our surprise, what we see is that upon neurodifferentiation, you can see that now there's a strong insulation that has formed at the promoter of this gene when it is not expressed, and there's also a strong contacts that are associated with the gene body. And this is independent of one of the known architectural proteins in the nucleus, which is CTCF, that can mediate such an effect, pointing to some novel biology. However, the major question that arises from those type of experiments is whether this is just a correlation or whether inducing gene expression can have a causative effect on genome topology. So we decided to address this question by employing the CRISPR-Cas9 system in order to artificially induce the gene on this location. So what we did is we activated the transcription using the dCas9 system, and you can see here that we achieved very strong levels of upregulation without any changes in cell fate. So now we had a system that allows us to test whether just inducing this gene will change the genome organization. And to somewhat of our surprise, it turned out that this is not sufficient. So inducing the expression of this gene basically didn't change the insulation level that we had at its promoter, and this is visualized here if you compare the actual experimental condition versus the control, and these are in the two stages of differentiation. So we think that there are other mechanisms in play, not just active transcription, for example, enhancer-promoter interactions, so these contacts over here might play a role in establishing such a topology. However, one other aspect that was very interesting is what happens at the longer ranges of genome interaction. So this is something that we were uniquely positioned in order to understand. And one of the things that we observed very surprisingly are these clusters of chromatin loops, essentially, that occurred sometimes over big distances in the genome, so more than 30 megabases apart. So if we zoom in even more on one of these regions, we can see that actually these kind of contacts are formed not between gene promoters, but actually between the genes themselves. You can see here that these are the genes. And another thing that we notice are that these are genes that are highly transcribed, and they contain multiple exons, as you can see, as you can see here by the RNA-seq. So we wanted to ask the question, is this specific to a few genes in the genome, or is it actually a phenomenon that occurs between multiple genes? So what we did is essentially we ordered the genes based on how many exons or how many splicing sites they have, and we asked whether they can form contacts in the nucleus or not. And what you see here is that only the genes with the highest number of splicing events essentially interacted inside the nucleus, while the genes that have few exons essentially did not interact. This phenomenon is also dependent on transcription because we see that it only happens when the genes are highly transcribed, and when the genes are switched off, they do not interact irrespective of how many exons they have. So this actually suggests that the transcription and splicing occurs, splicing occurs co-transcriptionally, and perhaps what we are seeing here are the nuclear speckles that have been speculated in the nucleus. So we are working currently on this. Another type of long-range contacts that exist in the nucleus that have been well described in the Drosophila system by Giacomo and also in embryonic stem cell by others are polycomb-based contacts. And polycomb is essentially a complex that is a repressive complex. And here what we see is indeed we can find a very strong context between two polycomb sites in embryonic stem cells. However, to our surprise, what we found is that upon differentiation, and particularly in the cortical neurons, you see that this context completely disappeared. So this suggests that despite the presence of the repressive mark mediated by polycomb, there's something else that is missing in there to establish this context. And we could verify these findings using a fish where we can label the two regions, and we see that only in the neurons, essentially, they come apart, suggesting that they're not contacting anymore. However, one of the major questions, especially for me as a neurobiologist in this study, was can neuronal transcription factors actually mediate genome organization? Is there something else that occurs? And indeed what we see is that we observed several transcription factors that are expressed in a precise sequence during neurodevelopment. So starting with PAC6, which is very highly expressed in neuroprogenitors, and ending in TBR1, which is a neuronal specific transcription factors, we asked the question whether the binding sites of the transcription factors come together in the nucleus in order to form 3D context. And what we found is that actually they do in a very cell type specific manner. So you see that PAC6, which again is expressed only in neuroprogenitors, mediates context between different regions in the genome, and you can see here that this occurs much more strongly in progenitors than in neurons. Conversely, TBR1, which is expressed in the opposite way, so mostly in neurons but not so much in progenitors, seems to mediate context only in the neurons. And if we look at what happens at the transcription factors that are expressed in a gradient, so in between, what you see is a rewiring, a gradual rewiring of the 3D genome organization from one mediated by PAC6 in neuroprogenitors to one mediated by NEURD2 in TBR1 in the neurons. And one of the things that we are currently exploring is whether the transcription factors have causative roles in this phenomenon, or whether they're actually following a predetermined genome organization. And here I just want to show you one example. Yes. So I want to, okay, so I'm going to skip the example, and I'm just going to show you one thing that we found about enhancers, because enhancers are very important regulatory elements. So we found that actually they interact with promoters only when the genes are active, and we found that this interaction is essentially very dynamic and changes drastically during differentiation. So to summarize, what I've showed you is that the genome organization represents a new, unexpected level of hierarchy together with transcription and linear marks, and determines cell fate in development. I showed you that active promoters can act as insulators, but transcription itself is not sufficient in order to induce this topology. And finally, I showed you that splicing contexts can form in all of the cell types, however polycomb contexts are disrupted, and we have different types of regulatory contexts, such as enhancer promoters, that are associated with transcription factors. So I wanted to thank all the members of the CAVALLI lab, which were really helpful in this project. Giacomo has been an incredible mentor in this and also in the lab. As Giacomo said, also Neta and Amos, who were very helpful. The Wellcome Trust for funding me, and I'm going to set up my own lab in the next couple of months, start at the Helmholtz Center meeting. So thank you for the attention, and I'm happy to take any questions.
Merci. Merci beaucoup. Donc la présentation est ouverte pour les questions. Oui.
Envelope or the nuclear pore complex, do they play any role in this rewiring?
We think that some of the specific regions are associated with the nuclear lamina. So these are mostly repressive type of domains that are close to the nuclear lamina. And we found actually that the repressive domains, essentially, they start to interact more strongly with each other during differentiation, especially in cortical neurons. Now, whether this is particularly related to the nuclear pore or it's more related to lamins, which are also changed in the development, this we don't know yet, and we continue to study.
Okay, I have a question. How do you measure your insulation score?
Right.
I mean, this Yeah.
So this is, it's actually quite simple because all we are measuring is how many contacts. So imagine you have a point here and we want to know the installation score of this point. So what we're measuring is how many contacts cross across this point. So if you have a position X and position Y, how many contacts do we observe between these 2 points? And how many contacts do we observe inside this region, so overlapping with the point? So for regions that act as strong insulators, you're going to have very few contacts that cross the certain region, right? So this is what we consider as an active insulators. For example, the gypsy insulator in Drosophila is one of the most famous ones, that if you insert it in a certain position in the genome, it would actually prevent contacts. It would prevent the surrounding regions from interacting with each other.
D'autres questions? I also have a naive question. So if I understand well, those domains are very reproducible from one cell to the other as long as they're in the same differentiated state, right?
Yes.
And but what was not completely clear to me is how it is functionally relevant, those domains.
Yes. So these domains have actually been shown to be very relevant because they're frequently dysregulated in human diseases. So in disease, for example, that's called polydactyly, that affects the number of digits, you have inversions or deletions in the non-coding part of the genome. And this inversion is actually across a domain boundary. So what it does is essentially it's an inversion that brings an enhancer, which is normally in a domain to the left, for example, to the other side. And this leads to an upregulation of a specific gene that then is responsible for the disease. So we have a very strong evidence that the topology of the genome is critical for diseases as well.
Thank you. So,
I have a question.
oh, okay.
So what is your most burning question that you are now addressing?
So yes, so the most burning question that I want to understand is how is the genome organization and transcription related in heterogeneous populations? So we all know also from the presentation by Valentin is that the brain is a very heterogeneous tissue. So we have many different types of neurons, as he showed, also many different types of synapses, and each of them has a distinct morphology, connectivity, and so on. So what I would really like to understand is first how genome organization is changing in individual cells, but also how does it influence evolution? Because, as you can imagine, the brain and the mouse brain and the human brain are very different, and together with the evolution of the genome at the linear sequence of the DNA, we also have an evolution of the 3D genome. And I think that this is a fascinating thing that deserves a study.
Okay, thank you very much.
Thank you.